Yeah.
There we go.
Do you want to use my computer?
Huh?
Yeah, maybe she should have a computer.
Do you want to use this?
Yeah.
No, how is it easier? Is it recording already?
It's recording.
Hello. Hyman's presentation. Take
You
one.
have enough room?
There's a bottle on the way.
10-minute video?
It should be okay. It's my phone. Yeah.
It's his phone.
Oh, it's his phone.
Yeah, it's my phone, so it should be okay. Alright, should I just start?
Yeah, start.
Okay. Everyone, thank you for coming. Thank you so much for your time. So my name is Hyman Tim, and I'll be presenting about the work I've been doing with Tarek for this past semester. So the project itself focuses mainly on Huntington's disease. Now, Huntington's disease is a inherited autosomal disorder that causes neuronal death and eventual cognitive deterioration, and it affects various parts of the brain. And the symptoms of Huntington has been shown to correlate with the areas that are affected. So, for example, if the caudate nucleus experiences deterioration, the patients can develop the onset of choreiform movements, which are the random jerky movements we see in Huntington's disease patients. So Huntington's disease itself is caused by a CAG trinucleotide repeat expansion within the gene Huntington, or exon 1 of the gene Huntington. And what this translates into is an expanded poly-Q tract within the protein. Normal proteins usually have around 10 to 35 residues, whereas the patients that develop Huntington's, or that patients that have fully penetrant Huntington, express more than 40 poly-Q repeats within the protein. So the function of Huntington itself, the protein Huntington itself, is unclear. We just know that it is essential for development. It is expressed in many cells, but it is shown to have the most activity within the brain. And the absence of Huntington has been shown to display lethality in mice. Whereas mutant Huntington disrupts many cellular processes that contribute to the pathogenesis of the disease. So some processes include the dysfunctional cytoskeleton, disrupted axonal transport, and disruption of protein clearance machinery. And also many of these pathways contribute to the pathogenesis of the disease. So Huntington's has a variable age of onset that has some correlation with the number of CAG repeats within the mutant gene. So if you can look on the graph here, we can see that a higher number of CAG repeats is associated with a younger age of onset. However, this correlation only accounts for a certain amount of the observed age of onset. And if we examine one specific repeat length, we can see that there is a variable age of onset even within that one repeat length, which means that there can be other factors to contribute to this age of onset, which leads to the hypothesis of our project, which states that if by using a cross-species and comparative transcriptomic approach, we can identify evolutionary conserved genes that have altered gene expression across different species. And in testing this hypothesis, we can gain further insight into the pathogenesis of Huntington's and identify novel therapeutic targets of the disease. Now, in order to do this, we took data sets from 3 different species: humans, mice, and Drosophila, and we looked at the genes that were dysregulated within these 3 species. And we selected certain genes that obeyed certain criteria, and these genes were significantly expressed within the models. These genes were evolutionarily conserved, and they're concordant in their direction, meaning that they're dysregulated in the same way, whether it was downregulated or upregulated. And examining these genes, a protein-protein interaction map was formed based on the directionality of the dysregulation. And if we examine the protein-protein interaction map of the downregulated genes, we can see that these genes are associated with synaptic transmission, cell signaling, metabolism, and nervous system development, which is something that we expect out of Huntington's disease. And if you compare this to the upregulated genes that
Why do we expect that?
Because Huntington's disease is a neurodegenerative disease. So, for example, dysregulation in synaptic transmission could dysregulate the nerve signals that is transmitted between the axons, and cell signaling, and similarly, and because nervous system development is, as I said, Huntington's disease causes the death of neurons. It could inhibit development of the nervous system. And again, if you look at the genes in the upregulated pathways, we see that these genes are involved in inflammation, cellular stress, cell division, and cancer-related pathways. So while this network analysis helps us identify the entities involved in the pathogenesis of Huntington's, it doesn't help us see any causal relationships. In order to find these causal relationships, we do our screens that we do in the lab. And these screens involve crossing flies and collecting their progeny and putting them through a test. And a big part of these fly crosses that we do, of course, is the GAL4 yeast system, which is taken from yeast. GAL4 is a transcription factor that binds to a site called UAS, which drives the expression of whatever transgene we pair with UAS. And we drive the expression of GAL4 using an endogenous gene within the flies. And for our screens, we use a gene called LLV. LLV is a gene that is expressed in all neurons specifically, and it is shown to be very significant in nervous system maintenance and development. And and then we pair certain transgenes with UAS. For example, we pair the expression of the mutant huntingtin protein and the shRNAs for our screens, for screening purposes of these flies. So what happens is we cross LIV and mutant huntingtin flies, which are the disease model flies, with flies that express certain shRNAs that are associated with the alleles that we want, of the genes that we want to test. So these crosses are made, and we collect the progeny and run climbing tests or motor performance assays through various stages of their life. And here LLV is very useful because it is, again, as I said, is a gene that expresses exclusively neurons, so it eliminates potential confounding factors within the assay that we perform. So what this test does is we use a machine, which is shown here, and what the machine does is, after we put the flies into vials, the machine taps the flies down to the bottom, and it films the flies coming to the top of the vials, and it measures certain parameters. And these parameters are compared to the controls that we put in, the controls we also test to see whether or not the genes that are silenced or are silenced using our shRNAs have an effect on the phenotype. And tests have shown that flies that express mutant huntingtin experience a decrease in their motor performance and their coordination. So one of the parameters, for example, that is tested is speed. As the graph shows, the control flies, which are flies that don't express the mutant huntingtins, demonstrate they basically move the fastest at the later stages of their life. Whereas flies that express Huntington's disease, or flies that express the mutant protein, have shown to have a decrease in their speed as they age. Now modifiers, depending on whether if they're a suppressor, they will show a phenotype that are somewhere between the control flies and the flies that express mutant huntingtins, and modifiers that are enhancers would show phenotypes that are worse than that of the flies that express mutant huntingtin protein. Now, in doing these screens, we can better categorize whether or not, or categorize the genes that are involved, genes that are dysregulated within the disease. So for example, if a certain gene is downregulated in Huntington's, we can test that gene through our screen to see whether that downregulation is a compensatory mechanism or a pathogenic mechanism. So for example, if the gene is downregulated and we further downregulate that gene using our shRNAs and the flies show a better phenotype, that gene could be classified as a suppressor, meaning that it suppresses the phenotype associated with Huntington's, or if a gene is downregulated and it shows a worse phenotype, we can classify that gene as pathogenic, meaning that it is part of that pathogenesis of Huntington's. So the screens have been continuing, and Tarek himself is still trying to gather more data about the recent screens that we've done, so this is not yet the completely updated data, but so far we've seen that the genes that we tested in the networks that have been created are very highly enriched in modifiers, genes that are seemingly actually involved within the pathogenesis of Huntington's. And continuing the screen will again help us identify important pathways within the disease pathogenesis, and will help us look at novel therapeutic targets of the disease. And looking forward, we will continue the screen, giving priority to the central genes, which are genes that are connected to or have a lot of connections in the protein-protein interaction map. We also cross these genes with the full-length model of Huntington's, which is a different model of Huntington's that we use in the lab, and we will test different alleles of the gene to better understand or get a better, broader picture of the role of these genes within the pathogenesis. And we will also create a humanized knock-in model of Huntington's, which means that we will try to substitute the exon 1 of the fly Huntington gene with the human exon 1 version, so that we can use the endogenous fly drivers for Huntington's to express the human mutant Huntington disease. And this is what I will be working on next semester. Yeah, so thank you, Tarek and Dr. Bodas, for helping me. And thank you so much for your time, and thank you for listening.
Thank you, that was very
is
good.
it. Thank you.